Hello everyone. In this video, we will introduce MSI team, which is one of the research teams in the Fukusaki laboratory. So here, MSI stands for Mass Spectrometry Imaging. And this video will explain the outline of mass spectrometry imaging and research results in the Fukusaki laboratory. Seeing is believing. This is the key phrase for MSI team. From a long time ago, researchers would like to see what we cannot see using new technologies. As the saying goes, seeing is believing. Visual information is intuitively understandable compared to complicated numerical information. One of conventional technologies is microscope. Using microscope, you can see structures, but you cannot find components. Imaging mass spectrometry is one of molecular imaging techniques using mass spectrometry. Using this technique, you can see many kinds of components without labeling. So, this is an overview of mass spectrometry imaging. Mass spectrometry imaging is a two-dimensional method of performing mass spectrometry on tissue sections. First, body tissues such as mouse brain and plants are sectioned. Molecules in the tissue are ionized by irradiating the tissue section with a laser for mass spectrometry. By repeating this on a two-dimensional tissue section, the position information and mass spectrum on each position can be obtained. Then, you can select the peak that appears at M over Z of the target compounds and obtain the distribution image of that compound. Mass spectrometry imaging allows us to know what kinds of substance, where, and how much exists from this image. Okay, this is an example of using mass spectrometry imaging on hot pepper. As you know, when you eat hot pepper, you can feel the spiciness of capsaicin, which is a pungent compound. However, it's impossible to know which part is pungent just by looking at the organizational structure of the capsicum as shown on the left of the photo. Therefore, we used mass spectrometry imaging to visualize the distribution of the capsaicin. Then, two peaks derived from capsaicin were observed in the mass spectrum. By selecting these peaks and generating images, we found that capsaicin was accumulating in the placenta rather than the pericarp. At Fukusaki Laboratory, IMSCOPE QT manufactured by Shimazu Corporation is used for mass spectrometry imaging. IMSCOPE QT is equipped with an optical microscope for observing the sample morphology and a laser used for ionization. Also, it can fuse with a mass spectrometer that measures the mass of ionized molecules. Then, it is possible to superimpose and compare optical images and imaging results. Our research team is trying to solve many biological problems using mass spectrometry imaging. Our research target is very wide. For example, in medical research, we target metabolites of cancer tissues and neurotransmitters in mouse brain. In food research, target includes not only fruits such as banana, but also processed foods such as rice koji and crude drugs. In addition, we also try to visualize microscopic organisms such as Drosophila melanogaster. And at Fukusaki Laboratory, we're working on the development of applications and methods for a wide range of samples other than those listed here. So, from now, I'd like to introduce recent research achievements. The first is the development of a method for visualizing enzyme reaction on plant tissues in MSI. The localization of enzyme reaction in living organisms is important for understanding biological phenomena. Enzyme histochemistry has been used to study the localization of enzymes. However, there are many limitations such as the need for staining, and also there are a limited number of enzymes that can be examined. Therefore, by introducing mass spectrometry on the tissue, we could compensate for these shortcomings and have succeeded in developing a new enzyme histochemistry method. In this study, we found high JAD activity in the vascular system in the hypocotyl and root tip meristem of germinating soybean seeds, which are also known as model plants. The next is enzyme activity imaging of insects. Many pesticides are used in the world, and they can have effects on more than just pests. However, whether neonicotinoids affect the distribution of enzyme activity in non-target organisms has not been investigated. Therefore, we visualized the effects of neonicotinoids on the distribution of enzyme activity by mass spectrometry imaging using the monomer organism Rhizophylla melanogaster. The analysis revealed that neonicotinoids inhibit enzyme activity in the fly hat. Okay, this is the third one, which is metabolite imaging of rice koji. Koji is an important ingredient in the production of sake, and it's said that the quality of koji greatly affects the quality of sake. The quality of koji is usually judged by observing the structure. However, little is understood about the metabolites that affect the surface structure. So that's why it's important to reveal the relationship between the quality of koji and its metabolites. It's also necessary to prepare section for visualization of koji, but it's difficult to make cross-section because koji is small and fragile. In addition, the analysis of koji using mass spectrometry imaging is an unprecedented attempt. Thereby, the purpose of this study is to develop mass spectrometry imaging method for metabolite imaging in rice koji. By using cryofilm, it has become possible to prepare section while maintaining the morphology of koji. Furthermore, we succeeded in maintaining ion intensity and localization by supplying a matrix by sublimation. Choline and lipophosphatidylcholine were discovered by MSMS analysis using this method and comparison with an online spectral database. And this year marks an 8th year of the MSI team. Many papers were published in 2022. So if you are interested, please feel free to contact us.